Chapter 14. Changes in Bioinformation. Change is imminent and inevitable. Everything changes, and so does genetic information. The change in genetic information is then reflected in the change in the phenotype, trait, or character. The consequence of the change could be neutral, bad, or good. There is no general direction. That change in genetic information is the basis for the variation in organisms. Those organisms that have better phenotypes would survive, while others might perish. That is natural selection. Change in the genome and selection is the essence of evolution. This subject matter is complex and needs finer details to understand comprehensively. I would suggest referring to any standard textbook. Here, we shall discuss only the changes in genetic information over time. Any change in the genetic information base sequence is a mutation. Causes of mutations. Mutations arise due to diverse factors, broadly categorized as replication errors, DNA damage from chemical or physical agents, and recombination involving endogenous or exogenous genetic material. Replication errors are inherent to the cellular machinery, while damage is often induced by environmental factors such as reactive oxygen species, ROS, chemical agents, and radiation. Recombination events involving exogenous DNA, such as those from plasmids, transposons, or viruses, further contribute to the mutation landscape. Mutations typically initiate as mismatches or structural damage to DNA, such as the loss or gain of functional groups in nucleotides. When these lesions evade repair mechanisms and are subsequently copied during DNA replication, they become permanent alterations in the genetic code. Mismatches due to replication errors. DNA replication is a highly accurate process facilitated by DNA-dependent DNA polymerases that synthesize a complementary strand using a pre-existing template. These enzymes depend on the principles of Watson-Crick base pairing to ensure fidelity. However, replication errors occasionally occur, resulting in mismatched bases. DNA polymerases possess proofreading activity to correct such errors, and additional repair mechanisms, such as the mismatch repair pathway, further enhance accuracy. Despite these safeguards, some errors persist, leading to mutations. For example, a common replication error involves mispairing thymine with guanine instead of adenine, resulting in a transition mutation. Another mechanism, replication slippage, is prevalent in repetitive DNA sequences and leads to the insertion or deletion of nucleotide repeats. This phenomenon underpins trinucleotide repeat expansion disorders, such as Huntington's disease, where excessive CAG repeats in the HTT gene cause progressive neurodegeneration. DNA damage as a source of mutations. The genomic integrity of organisms is constantly challenged by endogenous and exogenous agents that damage DNA. Endogenous factors include metabolic byproducts like ROS, which can oxidize DNA bases, while exogenous factors encompass chemical agents and physical stressors such as ultraviolet UV and ionizing radiation. These agents induce a variety of lesions, including the addition or removal of functional groups, the formation of covalent bonds between adjacent bases, and the breaking of the sugar phosphate backbone. Damage can manifest as single-strand or double-strand breaks, with the latter posing a greater threat to genomic stability. Chemical agents like alkylating compounds modify DNA bases, altering their pairing properties. For instance, methylation of guanine by methylmethane sulfonate, MMS, creates O6-methylguanine, which mispairs with thymine, leading to GC to AT transitions. Similarly, deamination of cytosine results in uracil, which pairs with adenine instead of guanine, causing CG to TA mutations. Physical agents, particularly UV radiation, induce the formation of thymine dimers, covalent linkages between adjacent thymine bases. These distort the DNA helix, obstruct replication and transcription, and, if left unrepaired, result in mutations. UV-induced mutations are strongly associated with skin cancers such as melanoma, where cumulative damage activates oncogenes or inactivates tumor suppressor genes like p53. Recombination and exogenous genetic material recombination errors, especially those involving exogenous genetic material, represent another major source of mutations. Endogenous DNA or external agents like viruses, transposons, and plasmids can integrate into the genome, causing genetic instability. Viral DNA integration, for example, often disrupts host gene function. Human papillomavirus, HPV, integrates its genome into host cells, inactivating tumor suppressor genes such as p53 and RB, a process linked to the development of cervical cancer. Similarly, retroviruses like HIV introduce genetic material that contributes to chromosomal rearrangements or gene disruptions. Transposons, or jumping genes, are mobile genetic elements capable of relocating within the genome. Their movement can lead to insertions, deletions, or rearrangements. For example, hemophilia A has been linked to transposon-mediated disruption of the factor VIII gene. During meiosis, errors in homologous recombination can lead to large-scale chromosomal aberrations, such as translocations, inversions, and duplications. The Philadelphia chromosome, a hallmark of chronic myeloid leukemia, CML, arises from a translocation between chromosomes 9 and 22, resulting in the oncogenic BCR-ABL fusion protein. Similarly, unequal crossing over can lead to gene duplications or deletions, as seen in Charcot-Marie-Tooth disease, where duplication of the PMP22 gene causes peripheral neuropathy. Replication before repair. The establishment of mutations often depends on whether DNA replication occurs before damage is repaired. Unrepaired damage can be erroneously copied, making the mutation permanent and heritable. Oxidative damage caused by ROS, for instance, generates 8-oxoguanine lesions, which pair with adenine instead of cytosine. This mispairing results in GC to TA transversions, which persist if replication precedes repair. Such mutations accumulate over time, contributing to aging and carcinogenesis. For example, oxidative stress-induced mutations are implicated in cancer development, where they affect key regulatory pathways controlling cell proliferation and apoptosis. Genetic changes are inevitable. Replication is a prerequisite for cell division, which leads to the formation of new cells with their own life processes. In some organisms, like bacteria, each round of replication and cell division results in 2 independent bacterial cells. However, in most multicellular organisms, including humans, replication and cell division give rise to new cells that belong to the same body. In these organisms, the body is divided into somatoplasm and germplasm. Only the germplasm is involved in the generation of new offspring, while the somatoplasm plays a role in supporting the survival, mate finding, and reproduction of the germplasm. All of the body's cells, except for the gametes, are considered somatoplasm, making up most of our body. Several rounds of cell division are required to generate trillions of cells that originate from a single cell, the zygote. Cell turnover in our somatoplasm means that cells die due to wear and tear and are replaced by new cells. This leads to an increased number of replications and cell divisions. As mentioned earlier, each round of replication can induce mutations. Therefore, the number of replications is directly proportional to the number of mutations. The human body exhibits mosaicism, where cells within the same individual harbor genetic differences arising from replication errors. Spontaneous mutations and environmental factors accumulated over time. These genetic variations occur during cell divisions, leading to somatic mutations that can result in distinct cellular lineages, influencing development, aging, and disease susceptibility. Most mutations do not affect the phenotype, but repair systems and homologous recombination can correct many. However, some mutations occur and can result in diseases like cancer. The germplasm begins to form during embryonic development, but remains inactive until puberty. It is intriguing to contemplate why the germplasm remains dormant for such a long period of time. In plants and many other organisms, there is a delay in fruiting. The division between somatoplasm and germplasm in plants is not as distinct as it is in humans. The gametes in plants arise from meristems, which are responsible for giving rise to other plant parts. Recombination, combining genetic information from 2 different DNA molecules in recombination. A standard textbook called Molecular Biology of the Gene, co-authored by Watson, starts the lesson on recombination as follows. All DNA is recombinant DNA. Homologous recombination is a fundamental process that involves the exchange of genetic material between 2 or more DNA molecules with non-identical genetic information. This phenomenon results in the creation of novel genetic combinations. Homologous recombination plays a crucial role in the repair of small-scale mutations and maintains genomic integrity. When a small-scale mutation, such as a point mutation or a single base insertion or deletion, occurs in the DNA sequence, homologous recombination can act as a corrective mechanism. The homologous recombination process requires both the target DNA, which contains the mutation, and an incoming DNA molecule with similar or identical sequences. The similar sequences in the incoming DNA act as a template for the repair process. The key steps of homologous recombination involve recognizing and aligning homologous sequences, exchanging genetic material between the DNA molecules, and resolving the recombination intermediate. During homologous recombination, the cell machinery identifies regions of similarity between the target DNA and the incoming genetic information. These regions are often flanked by specific DNA sequences that serve as recognition sites for the recombination machinery. The exchange of genetic material can restore the original unmutated sequence, effectively reverting the small-scale mutation. This process contributes to the fidelity of the genetic material over successive generations. It is important to note that while homologous recombination can correct small-scale mutations, it can also contribute to genetic variation by shuffling genetic material between homologous chromosomes during processes like meiosis. Diploidy plays a crucial role in maintaining genetic integrity by providing two sets of chromosomes, one from each parent. This redundancy allows for the repair of damaged genetic material through recombination and repair mechanisms during meiosis. Furthermore, diploidy ensures the stability of genetic traits across generations, preserving species identity. Repair, resistance to change. The repair mechanisms in genetic mutations are crucial in preserving genome integrity. They are resilient against forces that can cause changes in DNA sequence. These mechanisms, mediated by various repair pathways, ensure genetic information accuracy. In this article, we will explore the main DNA repair mechanisms. 1. Mismatch repair. Mismatch repair targets errors in base pairing that may occur during DNA replication. DNA polymerases usually proofread and correct mistakes, but sometimes errors persist. Mismatch repair detects and corrects these mismatches, preventing mutations. This process involves recognizing and removing mispaired bases, followed by correct DNA sequence resynthesis. 2. Nucleotide excision repair in ER. Nucleotide excision repair deals with a wide range of DNA damage, particularly damage caused by bulky chemical adducts or UV-induced thymine dimers. The damaged DNA segment is excised and replaced with correct nucleotides. This repair pathway is essential for protecting the genome from environmental damage. Base excision repair, BER. Base excision repair specializes in repairing DNA damage that involves the removal of specific damaged bases. It targets small, non-helix distorting lesions, such as those caused by oxidation or alkylation. BER involves specific enzymes that recognize and remove the damaged base, followed by the insertion of the correct base to restore the DNA sequence. 4. Global repair, SOS response. The SOS response is a comprehensive and coordinated cellular reaction to extensive DNA damage. When the frequency of DNA damage exceeds the capacity of conventional repair mechanisms, the SOS response is activated. This system induces an error-prone DNA polymerase, allowing for DNA synthesis even in the presence of damaged templates. Although this mechanism is error-prone, it is considered a last resort, emphasizing the something is better than nothing approach to DNA repair. Its main goal is to prevent the collapse of cellular functions in extreme conditions. DNA repair systems have evolved as remarkable safeguards to preserve genetic stability by detecting and correcting errors or damage, thereby reducing change. A striking example is fail-safe glycosylase, a repair mechanism targeting oxidized bases like 8-oxoguanine, which can pair incorrectly with adenine. Of the OXO-G and adenine, OXO-G is the damaged nucleotide, and adenine is the normal base. Yet a special glycosylase removes adenine that is base paired with OXO-G. Another marvel is the mismatch repair system, which corrects replication errors by distinguishing the newly synthesized DNA strand from the parent strand. This distinction relies on transient methylation patterns, where the parent strand is methylated, guiding enzymes such as MUTS, MUD-L, and MUD-H to excise and repair the mismatched region. These mechanisms illustrate the extraordinary evolution of repair pathways that maintain genomic integrity, demonstrating nature's precision in reducing harmful changes. These are examples of impressive links the evolution has come up with phenomena to maintain genetic integrity. Notice that all the systems are an effort for the existence of genetic information, not its evolution. Several factors influence the rate of genetic change. Spontaneous mutations accumulate over time as the duration of an organism's existence increases, providing more opportunities for genetic change to occur. The number of replications is also a key factor, as replication errors are proportional to the number of times a cell divides. With more replications, there is a higher likelihood of introducing errors. Additionally, environmental agents such as chemicals, physical forces, and biological agents can further increase the rate of genetic change. However, the balance between genetic change and stability is maintained by repair mechanisms, which correct errors in DNA, and homologous recombination, which can lead to insertions and deletions depending on the specific molecular events. The rate of natural mutagenesis is influenced by the number of replications and the number of functional repair genes, with higher repair capacity reducing the rate of mutations. To derive a formula for the rate of spontaneous mutagenesis, we can integrate the key factors influencing genetic change: the duration of existence, the number of replications, the influence of environmental agents, and the contribution of repair mechanisms. The rate of spontaneous mutagenesis, R, can be represented as formula R equals KTNR1 NRP 1 plus ER. Rate of spontaneous mutagenesis, mutations per unit time, which is dependent on the fidelity of the enzyme and the efficiency of the mismatch repair system. Also, the damage is caused by metabolic end products and the other repair systems. Baseline mutation rate per replication, which depends on the fidelity of DNA polymerase and other cellular factors. T, duration of existence, time or lifespan of the organism or cell. Number of replications or cell divisions during T, NRP. Number of functional DNA repair genes or repair efficiency. Higher NRP reduces R, reflecting the corrective capacity of repair systems. Contribution of environmental agents to mutation rate. It is dimensionless and normalized, where equals 0 represents no exposure to external mutagens, and E0 represents increasing exposure levels. Duration of existence T. Mutations accumulate over time, as each replication cycle presents opportunities for spontaneous errors. Longer durations increase the total opportunities for mutations. Number of replications NR. Mutagenesis is strongly influenced by the number of replication events, as each division introduces a chance for errors to occur. Organisms or cells undergoing frequent division accumulate mutations at a higher rate. DNA repair efficiency, NRP. The number or efficiency of DNA repair genes inversely impacts the mutation rate. A greater number of repair genes improves error correction, reducing the accumulation of mutations. Environmental influence, E. While this formula primarily addresses spontaneous mutagenesis, E accounts for the influence of environmental agents. For environments free of mutagens, E equals 0, but in mutagenic environments, E0 amplifies the mutation rate. Baseline mutation rate, K. This is an inherent property of the organism or system, determined by the accuracy of DNA replication machinery and intrinsic cellular conditions. The formula balances the mutagenic factors, time, replication, environmental exposure, against stabilizing factors, repair efficiency. It captures the cumulative nature of mutations over time and highlights the critical role of repair mechanisms in reducing mutation rates. The environmental term, E, provides flexibility to extend the model to account for mutagen-induced variations. Simplifications or extensions. For scenarios with negligible environmental agents, the term 1 plus E can be approximated as 1. If the focus is purely on replication and repair dynamics, R equals KTNR-NRP provides a simpler model. The rate of spontaneous mutagenesis, as predicted by the formula, reveals intriguing patterns in mutation rates across diverse organisms. Several factors, such as the duration of existence, the number of replications, and the efficiency of repair mechanisms, shape these rates. The number of replications is critical in determining mutation rates. Organisms with high reproductive rates, such as insects, undergo numerous replications in a single generation, leading to a higher probability of replication errors. Conversely, organisms with lower replication rates, such as slow-growing plants or larger mammals, will experience fewer mutations over the same period. The effectiveness of repair mechanisms also influences mutation rates. Organisms with robust DNA repair systems, such as certain yeast strains and vertebrates, may maintain a lower mutation rate even with frequent replication errors. On the other hand, organisms exposed to environmental stressors like UV radiation or toxins, or those with less efficient repair mechanisms, are more prone to accumulating mutations. Mutations in gain-of-function in cell cycle promoters or loss of function in repair genes can significantly impact the rate of spontaneous mutagenesis, accelerating carcinogenesis. According to the formula mentioned above, mutations in these genes increase the number of replications, NR, leading to more opportunities for replication errors. Simultaneously, impaired repair mechanisms, NRP, reduce the correction of these errors. The result is an increased mutation rate, R, driving the rapid accumulation of genetic changes and facilitating cancer progression. Carcinogenesis could manifest more quickly in such conditions due to the amplified rate of mutations. Somatoplasm undergoes more replications than germplasm, leading to a higher accumulation of mutations in somatic tissues than in germline cells. Germplasm remains highly conserved to ensure genetic stability across generations, which is likely why species with prolonged dormancy periods experience fewer mutations. During dormancy, cellular processes slow down, reducing replication errors. In contrast, somatic cells are more prone to accumulate mutations over time due to frequent replications. The size of an organism also affects its mutation rate. Larger animals, which produce more cells over their lifetimes, are expected to accumulate more mutations due to the higher frequency of replications. However, despite their large size and numerous replications, species such as elephants and whales exhibit low rates of cancer and age-related diseases. This suggests that large animals have evolved highly efficient DNA repair mechanisms or cellular systems that can counterbalance the increased mutation rates. Similarly, perennial trees, which live for many decades or centuries, undergo numerous replications but show minimal negative effects from mutations. This could be attributed to the high fidelity of their replication machinery or efficient DNA repair systems, allowing them to endure extensive replication without suffering from the typical consequences of genetic instability. Genome and diversification. A hypothetical genetic system with 100 genes, each combinable with 100 promoters, 100 operators, and 100 enhancers, results in 10 to 8 unique gene modules. A genome containing 50 to 25,000 such modules creates an immense number of possible configurations. This staggering diversity allows for vast regulatory landscapes, where differences in promoter strength, operator interactions, and enhancer effects alter gene expression. Such variation is a fundamental force driving evolution, as even minor regulatory shifts can lead to significant phenotypic changes. However, most combinations will be non-viable, as they may disrupt essential pathways, cause imbalances, or lead to toxic interactions. Only a small fraction will achieve a functional balance, forming viable genomes that can survive and adapt. Natural selection ensures that advantageous configurations persist while dysfunctional ones perish. Beyond genetic variation, epigenetic regulation further enhances diversification. DNA methylation, histone modifications, and chromatin remodeling influence gene accessibility, allowing identical genetic sequences to yield distinct phenotypes. These mechanisms provide plasticity, enabling rapid adaptation without altering the genetic code. Over time, stable epigenetic modifications may become heritable, contributing to long-term evolutionary diversification.